Hi everyone, and welcome to part one of this series where I'm going to share with you a few tools that can help you to get better insights into your membrane proteins during protein production and characterization. Today I'm going to focus specifically on protein production. So, let's face it, working with membrane proteins is really difficult. And I'm guessing that a lot of you have struggled with finding the right expression and purification conditions, for getting folded membrane proteins, and then finding the best detergents or lipids to stabilize your membrane proteins for use downstream, like for structural and functional studies. Now, I've experienced these same challenges firsthand because I worked with membrane proteins for over 7 years during my PhD and postdoc, which is why I want to share some of the tools that I wished had been around then, because it would have made my work so much easier, and ultimately I probably could have got a lot further with my research. Now, I worked on a protein called the 18 kilodalton translocated protein. And what I was trying to do was measure, trying to build a model membrane system with this protein embedded, and then study its structure and binding to potential ligands. So I did a lot of the more traditional radioligand binding experiments using cell lysate, but the huge challenge came when trying to overexpress and purify this membrane protein, and especially in a way that was folded and functional. So like you, I had to find suitable expression conditions, and especially ones that didn't result in inclusion bodies. Then, find a detergent that could extract and solubilize the protein, but without completely unfolding it. And then, since my aim was also to reconstitute it into model lipid membranes, I then also had to find lipid conditions that would enable me to study binding to what was, I hoped was a folded and functional protein. So as I'm sure you're all too aware, all of this is largely a process of trial and error to find the best conditions. And it can be a really tedious process, really laborious, and not to mention it can get quite expensive when it comes to the detergents and lipids that you need to test. So today what I'm going to focus on is a couple of technologies that I think can really help to make your work a lot easier and to help you to overcome the challenges in producing folded and functional membrane proteins. Now, as I mentioned, the first hurdle you've likely faced is finding the right constructs and expression systems, expression conditions for producing recombinant membrane proteins that are correctly folded, have a decent yield, and are stable enough to use for your downstream applications. There are now some in silico methods, that will help you to screen and identify potentially stabilizing point mutations for a membrane protein. But ultimately, you're still going to have to test these different constructs and expression conditions to find out which ones actually yield usable membrane proteins. Now, an STS page gel, it will tell you which expression conditions result in the highest yield of your recombinant protein, but it won't tell you whether that protein is properly folded or functional. If we first look at the STS page gel in this example, you can see that the overall yield of this target protein is highest when the E. coli is grown at 25 degrees rather than 16 or 20. But as you'll see, this isn't actually the full picture. Now, what you can see in the top graph is the unfolding profiles for the recombinant protein grown under the same conditions shown in the gel, but measured with the Andromeda instrument. So what the Andromeda is, it's the latest tool that allows you to screen lysates for the presence of folded recombinant protein to help you identify the best expression conditions for your membrane protein. Now, it measures thermal unfolding of your target protein in crude lysate by detecting changes in fluorescence, and this can either be from a site-specific his-tag dye for his-tagged proteins or from GFP fusion proteins. Now, in this graph, these peaks show that there is folded protein present in each lysate, and the height of the peak corresponds to the amount of the folded protein in each sample. So what you can see is that actually there's a higher yield of folded protein when the E. coli is grown at 16 degrees rather than 25. So that's different to what the gel is saying, so showing you that the conditions with the highest overall yield of protein, they don't necessarily correlate with the highest yield of folded protein. So you can see how the andrometer not only tells you about the expression levels of your target protein under different conditions, but also whether or not you are expressing folded protein. Andrometer will also allow you to monitor the effects of ligands on the stability of your membrane protein. That's in the same assay by performing a thermal shift assay. So what you can see here is the unfolding profile for a recombinant membrane protein in lysate, and that's both with and without a ligand. And you can see that with the ligand, it increases the unfolding temperature of the protein by about 10 degrees. So within the same assay, you can confirm the presence of folded recombinant protein in the lysate, from various expression conditions. And you can also see whether or not it's likely to be a functional protein that binds its ligands. So overall, that should really save you quite a lot of time in finding the right expression conditions to make good quality membrane proteins. Hopefully these examples have given you a good taste of how Andrometer, will let you quickly and easily screen different expression conditions before full protein purification, and to help you identify which ones will produce the most stable and likely functional membrane protein. So with Andrometer, you can measure up to 48 samples in 11 minutes, and that's in crude lysates over a wide concentration range. So I know that I haven't gone into a lot of detail here about exactly how it works, but if you would like to learn more, there's a link to the brochure in the resources, or of course you can reach out to us at Nanotemper at any time. As I'm sure you know, finding the best expression condition to get recombinant membrane protein is just the start. And the next challenge you face is finding a way to extract, solubilize, and purify your membrane protein. Detergents are still the main way that, the main thing we use to extract and purify membrane proteins, but it is always a balancing act to find the most suitable detergent, that's strong enough to solubilize the protein, but also gentle enough to keep it folded. So once again, there's a lot of trial and error in finding the best detergents for each membrane protein. Now, finding the right detergent to stabilize your membrane protein, as you're probably aware, is a bottleneck for structural or functional studies. And a lot of the techniques for measuring protein stability often require you to use dye, or they might be incompatible with detergents. So to overcome this problem, these researchers, they've established a high throughput workflow to screen the stability of membrane proteins. And in this case, it was 9 different membrane proteins, each screened against 94 different detergents. Now, first, each membrane protein was purified into DDM as a starting point, and then it was diluted into an excess of each of the new detergents to be screened. Then the unfolding and the aggregation of each membrane protein in each of the various detergents was measured with the Prometheus instrument. So what the Prometheus does is it will simultaneously measure the thermal unfolding and aggregation of a protein using the intrinsic fluorescence of that protein. You can see in the graph that the unfolding temperature was quite different depending on which detergent it was in. And in the table, you can see the melting temperature and onset of aggregation temperature of the protein in each of the various detergents. And you can then see whether these were increased or decreased compared to the DDM. So using this method, they could screen all 94 detergents for one membrane protein within just a few hours. And that was without needing to do a buffer exchange or without needing to use any dye. So you can see how the Prometheus can help you to find the best detergents to stabilize your membrane protein. And most of the time, you're likely using detergents to extract your membrane protein during purification. That's unless you're using something like SMELPS for extraction. But as we know, detergents typically strip the surrounding lipids from a membrane protein during solubilization. But now we know a lot more about how this can negatively impact membrane protein structure and ultimately function. Because the native lipids, they can be really crucial for the proper membrane protein conformation and function. So, you might be looking for ways to introduce or reintroduce lipids to your purified membrane proteins to help restore that native conformation and function. Again, finding lipids that will increase the stability of your membrane protein can be very time consuming. And it can be, quite expensive, especially if you have to purchase many of the different types of lipids. So to help with this, these researchers actually developed a lipid screen to be used with the Prometheus, and they tested membrane protein stability in 23 unique lipids and lipid mixtures. Now you can see in these graphs the unfolding curve of the adenosine A2A receptor in detergent and then in various lipids or with a ligand. Now cholesterol is known to be important for the conformational stability of GPCRs. And so here you can actually see that this was confirmed with the Prometheus. So the addition of the soluble form of cholesterol, that's the CHS, it resulted in a 10-degree or so increase in the melting temperature. Now, if you're looking for a way to help, to restore your membrane protein to a more native conformation, you can see how the Prometheus can really help to save you time and probably money when it comes to searching for relipidation conditions. So just to recap, the Prometheus can simultaneously measure protein unfolding and aggregation of your membrane proteins, and that's using intrinsic protein fluorescence, which means it's also compatible with any buffers or detergents, lipids, or other additives. So the andrometer, that can be used to measure protein stability in crude lysates earlier in your workflow, and that's to help you find the best expression conditions for your membrane protein. And then the Prometheus can be used for label-free measurements to find the best detergents and lipids for your purified membrane proteins. Again, you can find more detailed information about how the Prometheus works in the resources. So thanks for joining me today. Hopefully you've got a good overview now of how Prometheus Andrometer can help, make your work a lot easier when it comes to expressing, purifying, and stabilizing your membrane proteins.